Hello, everyone. I'm Xiukun Wang, a postdoctoral fellow from Guanghu's lab at NIEHS. It's my great honor to share my research work in cell regeneration, the OS talk. Today, I want to talk about the topic of blastoids, about how to research on early development using embryos in a dish. In January, we published one paper entitled Human Embryos in a Dish, Modeling Early Embryonic Development with Pluripotent Stem Cells, highlighting the newly published Nature paper about human blastoids, model blastocyst development and implantation. Since we are talking about blastoids, what are blastoids? In general, blastoids are mouse or human pluripotent stem cells self-organized into blastocyst-like structures. Literally, a blastoid is an embryoid, a stem cell-based embryo model which morphologically, transcriptionally, and functionally resembles the early pre-implantation development. Now that there are so many similarities between blastoids and blastocysts, it's necessary to introduce some background about blastocysts. Blastocyst that we are familiar with is a structure formed in the early development of mammals. Mammalian embryogenesis is initiated by gametes fertilization to form pluripotent zygotes. Once fertilized, the zygote undergoes several cleavages with no significant growth and cellular differentiation. It will lead to the development of a multicellular blastocyst until differentiation to hundreds of cell types to form a complete body. At the transition between pre-implantation and post-implantation, the blastocyst then compact, polarize, and undergo the first two lineage segregations to generate three different cell types. The first lineage segregation specifies an outer tropho ectoderm layer, TE layer, and an inner cell mass, SAM. SAM cells later segregate into epiblast, EPI, and primitive endoderm, PE lineages. Epiblast cells eventually give rise to all the tissues of the future fetus, whereas the PE gives rise to extra-embryonic endoderm cells that will form the yolk sac. Pre-implantation and development seems largely conserved between mice and humans. However, post-implantation may differ whatever in the structure and molecular underpinnings. At E5.5 to E6 day, once blastocysts have developed to the egg cylinder stage to form a cup-like egg cylinder. Instead of forming a cup-like egg cylinder in the mouse embryo, epiblast cells in the human or primate embryos are flattened to form a bilaminar disc at E10. That's the big difference between mouse and human post-implantation development, at least in the morphology. Referring the structural difference between mice and humans inspired us to employ different induction strategies to generate blastoids. Fortunately, stem cell lines from three different lineages, the TE and the PE, and also pluripotent epiblast, EPI, within blastocyst have been derived and stably maintained in vitro. TS cells are derived from the TE, XEN cells from the PE, and ES cells from the epi. Mouse embryonic stem cells are first derived by Martin Evans' lab in the 1981. Then the different states, primed and naive states of human embryonic stem cell lines are established in the 1998 and 2010 respectively. Trophoblast stem cell line TSC and extra-embryonic endoderm stem cell line XEN cells have already been generated successfully by docent groups and some other labs. Taking the in vitro cultured cell lines, we have great understanding of genetic and epigenetic regulation and also signaling pathways underlying mouse and human early embryonic development. But unfortunately, all the findings based on established cell lines are still plain and local, not interconnected and holistic. As an alternative, stem cell-based embryo models may open an unprecedented avenue for modeling embryogenesis. Understanding embryonic development can provide important insights into both basic research and clinical applications. However, it's difficult to study the natural development of mouse and human embryos for technical and ethnic reasons. In mice, it's a little easier to harvest the embryos, but still there are 3 hours to follow. While in humans, because of the 14-day rule, the supply of human embryos is extremely limited for ethnic issues, but it's also challenging to carry out experiments in vitro due to the lack of embryonic materials. Thereby, it needs to preclude the process of mechanistic understanding of the early embryogenesis in human. To systematically interrogate early development, a robust in vitro model of mouse and human embryogenesis is urgently needed. Besides the urgent need for research, the success of derivations of different cell lineages provides the foundations for blastoid generations. For these 2 reasons, the blastoids are coming into being. The cell lineage specification and the basic principles underlying embryonic development have not been pursued by scientists. Blastoids possess distinguished properties that promote the research or development biology. Actually, the concept of blastoids is evolving along with time. Early in 1980s, Evans and Martin have taken firstly derived mouse embryonic stem cells to form embryoid bodies termed EB. This structure is first known to us, showing embryonic stem cells can differentiate into germ lineages within 3D embryoid bodies. Then, a great breakthrough is made by Magdalena Gold's group. They found that mouse pluripotent stem cells can be induced to undertake a similar process of self-organization into rosette structure, following their in vitro culture suspended in the extracellular matrix proteins. It's the first way that we realized in proper culture conditions, the pluripotent stem cells possess self-organization property to initiate morphogenesis in vitro. Eventually, until 2018, Nicholas Reverend unprecedented claimed the concept of the blastoids. They report that trophoblast and embryonic stem cells cooperate in vitro to form structures that morphologically and transcriptionally resemble E3.5 blatocysts. Following this report, several other labs have reported modified methods for blastoid generation that all recapitulate the blastocysts' overall morphology and contain the 3 distinct cell lineages. Only 4 years since it was proposed, actually blastoids experienced 40 years of evolutions and development. Here, I will share some key events that happened in mouse and human blastoids. As I said, the Magdalena Gold's group firstly raised the self-organization concept to show the self-organization properties of mouse pluripotent stem cells initiate morphogenesis upon implantation. After that, he also compared embryonic and extra-embryonic stem cells assembly to mimic embryogenesis in vitro. Until 2018, the first blastoid was formed using embryonic stem cells cultured in the ground state condition in order to form epi and PE combined with the multipotent TSC to mimic the TE. In contrast to the protocols based on mouse embryonic stem cells, totally 4 groups reported blastoids based on marine EPSC, expanded pluripotent stem cells without the addition of the TSC to the generation of EPSC-only blastoids. Although EPSC-only blastoids can form blastoids with few exogenous clues than the other models, the efficiency of blastoid generation appears higher when they using TSC for the TE compartment rather than TE induction from EPSC alone. Following the report, several other labs have reported modified methods for blastoid generation that all recapitulate the blatocyst morphology and contain the 3 distinct cell lineages using 3 types of mouse blastocyst-derived stem cell